Yeah, anyway, thank you to Shitabra and Shakti and the other organizers for inviting me. It's really nice to be here. So, I mean, I thought, as Shitabra said, that because this is a workshop on games, I should talk to you about some work in which we use game theory. We actually use a combination. What I want to show you is a little bit of how we use a combination of nonlinear dynamics and game theory in a particular setting. And the setting is in this context of these viruses that infect bacteria, and they're called bacteriophage. Some of you would know about them, but I think many of you might also not know about bacteriophage. So I'll give a little primer about at least some of the important aspects of the dynamics of these bacteriophage and how they work. Can everyone hear me? Fine? Yeah, and please interrupt me anytime, whether it's about the mathematical modeling or about the biology of phage. Just feel free to interrupt. I will, I know this is a conference about games and networks, so I will try and bring in a little bit of networks also, depending on whether there's time, but it's not like I have a agenda of what I need to get through. So if you want to stop me, feel free. I'll be happy to just get through in a proper way, convey to you whatever I manage to convey. It's fine. It's not like I have to finish some particular set of topics. So let me begin by talking a little bit about bacteriophage. These have somehow always intrigued me as an organism. I mean, many of you must know that bacteria are very, very, very abundant on this planet and really shape a lot about the planet's climate and the biosphere and the evolution of everything on this planet. But some of you may not know just how much they themselves are influenced by these tiny little particles which are sort of on the edge of life and non-life, which are these viruses that infect bacteria. They are the most abundant predators of bacteria, and just as bacteria shape the evolution of the whole biosphere, they shape the evolution of bacteria. So they are an unusual predator-prey system. These are just some images which kind of show just how sci-fi looking these things are. They're about 10 times smaller, or order of magnitude, or 2 orders of magnitude smaller than a bacterium. And what they do is they attach onto bacteria and inject their DNA or RNA sometimes, but I'll talk mostly about DNA viruses. They inject their DNA inside the virus, inside the bacterium, and this DNA starts producing various proteins. It uses the machinery of the cell, of the bacterial cell, uses all the proteins and all the stuff that the bacterial cell uses to replicate its own DNA, to produce proteins, etc. It uses that to produce its own stuff and to replicate its own genome and eventually make lots of copies. So eventually what happens, so this is a little bit of an artificial situation where some experimentalist has exposed this poor bacterium to like 100 viruses. So these are these little virus particles all attached. And these viruses inject, and eventually they use the cell to make hundreds of copies, as you can see, of viruses, which will eventually burst the cell and come out. I mean, but this is a situation where this poor bacterium has been exposed to a really huge number of viruses, but it doesn't need to. Even a single virus can create a situation like this. A single virus genome gets in and replicates and can produce 100 new offspring. So just to give you an idea, in terms of like, we often talk about predator-prey systems like rabbits and foxes. So imagine there was a fox and it eats one rabbit and it produces hundreds of foxes from eating one rabbit. So this is the unusual aspect. The unusual aspect is, how you would imagine if that's the case, basically there should be no rabbits left, right? They should just be decimated. So how can bacteria and these bacteriophage even coexist? A single virus, yes?
In principle, can one virus infect an entire bacterium?
Yeah, if you think about successive rounds of infection, yes. So let's say a single virus finds a colony of bacteria, infects one bacteria, produces a few hundred phage, which then go on and infect, some of them will get lost, but let's say another 20 or 30 bacteria get infected, which produce equally another few hundred each, which then go on, and it can happen. And in fact, people do this kind of experiment in the lab. A very common experiment, and you may have heard of this, it's called plaque assays, is where you infect a lawn of bacteria on a petri dish with one, you can even do it with one, but let's say a very small number of viruses. And you see this little clearing happening in the where you've put the viruses, and the bacteria lawn is just cleared up and keeps all the bacteria dying, and it just grows circularly, and it will keep growing as long as the bacteria are growing. So indeed, a single virus in principle can infect a whole colony. So indeed, in some situations you will see complete extinction. So I mean, you can understand, given these numbers, it's kind of unusual. One second. It's kind of unusual, and it's a question of how these things can coexist. Yes.
You showed of multiple phages attacking one bacteria. Do we see some competition between the phages themselves?
I will come back to that a little bit later in the talk. In this case, it's all the same type of phage, so it doesn't really matter. One of the phage inserts its genome, and that genome will use the machinery and produce offspring, and they're all the same type of phage. But indeed, sometimes you can have multiple phages attacking, and there can be competition. Lots of games that way are possible. I will mostly stick to a single type of phage in this. I mean, maybe at some point, if there is time, I can talk about multiple phages. But already, even at the level of a single type of phage infecting bacteria, there's already some games that one can imagine. But I'll talk about a little bit more a little later in the talk. Okay, so one reason why they manage to coexist despite this unusual sort of predator-prey behavior is, of course, that bacteria also have defenses against infection. Many of you would have heard of CRISPR systems. CRISPR systems are bacterial defense systems against precisely these kind of viruses. But another reason why coexistence happens is the viruses, some class of these viruses called temperate bacteriophage, are self-limiting. They have the ability to do two things. They can either, so this is a bacteriophage, it inserts its genome into the bacterium. This is the bacterium, this is the bacterial genome. And two things can happen at this point. Either it can take over the machinery, just like I was talking about, and create lots of viral proteins and create more copies of the viral genome and eventually create more virus particles and burst open the cell. That's called the lytic cycle or lysis. But the other thing that can happen is it can go dormant. This genome of the virus can insert itself into the genome of the bacterium and pretty much live happily ever after. As the bacterium grows and divides, its genome duplicates and splits, and the viral genome also gets copied and split. So you can have this entire population of bacteria with this viral genome sitting dormantly inside it. And there are certain conditions in which it can jump out and come, but it's actually quite stable. So this is another way in which the virus, by going into a dormant state, can sort of stick around and not drive the bacterial population to extinction. And one of the things I've been very interested in for a long time has been, how is this decision made, what kind of information goes into this decision, etc.
Does this affect the bacteria in any way?
Not much. I mean, this happens, there is a very wide class of bacteriophage that does this. In some cases, we know that it makes no difference to the growth rate. In some cases, it may make a small dent on the growth rate, slightly reduce it. In some cases, there are even cases known where it actually enhances the growth rate because it has some gene which helps some metabolism in a particular kind of environment. So in certain environments, it could even help. So I mean, all sorts of things happens, but they are very minor effects. On the whole, in most cases, the bacterium pretty much grows at the same rate at which it grows without this virus. And in the wild, if you pick up bacteria from soil or marine situations and so on, it's very common to find at least 4 or 5 such virus genomes in bacterial genomes. That's so typically any bacteria in the wild will probably have such dormant things sitting inside it, of different species, not the same species. Okay.
Which branch will be taken? Is it a random process?
So that's the question. I mean, these are the kinds of questions that I'm interested in. Is it stochastic? If it is stochastic, is it nevertheless biased? It may not be 50-50. What bias is it? We know certainly that under certain situations, certain kinds of environmental conditions or certain kinds of growth conditions, it is more biased this way or that way. So how does this, another question is, how does this information about the environment get detected by a genome that's sitting inside a bacterium, and how does it decide, what are the genetic networks? So you can see there are a lot of questions one can ask about this kind of decision. And I mean, I'm not going to go through all these questions, but I just want to make the case that the questions can be asked at many different levels. You can ask the question about the gene networks that are inside, that are encoded by the genome. You can ask, what proteins are being produced by this virus? How do these proteins affect each other? What kind of feedback loops are there? How are they used to get information, incorporate information from the environment to make this decision? How is the decision made in the first place? You can ask at the level of the cell itself, what is happening with the decision? Is it like 50-50 like Sagar was saying? Is it 60-40? Is it 80-20? What decides how much dormancy happens, how much lytic virulent stuff happens? So this is actually the scale at which one can use a certain amount of game theory, and I will spend much of the talk talking about some questions we have investigated here. If I have time, maybe I might go back to the networksy stuff here. But a third level is of course the population level. What are the consequences of a decision process like this on patterns that you get? This I really won't have time to talk about, but I just want you to know there are many questions you can ask at all these levels. Okay, so before I get to this level and the game theory stuff and so on, I still want to give you a little more primer about what is actually happening inside a virus, in the viral genome and inside a infected bacteria, and some basic idea of the dynamics of the process. Okay, so this is what I said, that there are these two processes, and I said this is very stable. In fact, there is a certain sense in which even this state is very stable. On the face of it, it's not stable because you burst the cell and the cell dies. It's hardly a stable state. But people have in fact done experiments where they've taken mutants which stop this process somewhere along the way, and all of these mutants, if they go into this state, are very stable. So in some sense, if you discount the final bursting of the cell and the release of things, in some sense the internal network of this virus allows it to go into two stable states. So somewhere underlying in the genetic network of this virus, there is the possibility, there is a dynamical system, which in the language of dynamical systems, which you would have been heard in this workshop, is a bistable system. So we certainly know that it's capable of bistability. Yes?
Become immune to further attacks?
Sorry. Actually this is immune. This is called an immune state because this is not, although I said it's kind of dormant, it's not completely dormant. There is known to be in this state one particular viral protein that is produced, which kind of keeps all the other genes of the virus repressed. So what happens is when another virus of the same type or a very similar class infects this kind of bacterium and inserts its genome, that protein is already there and suppresses that genome also. So in effect, a new infection cannot take over this state. That's why it's called the immune state.
Has it been characterized that these two states are the only states that are possible? There's no intermediate?
As far as people can make out, they have not observed. I don't know if people have, systematically perturbed the system so much to try and do this. Lots of mutants have been looked at, and with the wild type system, there have been a few kinds of perturbations, and it seems like these are the only things happening. So it seems like there's a bistable system, and in the textbooks, yeah, sorry.
Single stranded or double stranded DNA?
In this case, we are talking about double stranded DNA. There are of course a lot of different kinds of viruses, also RNA viruses. I won't go into that. There also you have these kind of mechanisms, but sometimes there's a little extra complication when they're RNA or single stranded. But a very large class are double stranded DNA, so here it's all very clear. So the textbook picture, which has been around for a long while, especially for one of the very well studied such temperate bacteriophage, is something called lambda phage. I mean, it's been around for decades. If some of you know the history of molecular biology, this virus has been really at the heart of the development of molecular biology way back when it was Max Delbruck and others who were developing the whole field of molecular biology. It was what was used to understand very basics about how proteins can activate and repress each other and things like that. So since those days, there has been a sort of a textbook picture developed of lambda phage or other similar temperate bacteriophage about what is underlying this bistability. And the picture is there are two proteins. In the case of lambda, they're called C1 and CRO, and each protein inhibits the production of the other protein. Okay, So you can see automatically how this can produce bistability just by hand waving logic, by realizing that if you have a state in which C1 is high, it will repress CRO will be in low numbers, and therefore this inhibitory link, which is represented this way, won't act. So you'll have a state where C1 is high, CRO is low, and that's stable. Or you can have, since it's a symmetric sort of system, at least on the face of it written like this, you can have one where CRO is high and C1 is low, and that will also be stable. And indeed this kind of network structure is a very classic kind of switch which can produce bistability. And this has been, I mean, in lambda phage this is known. In this state, this is the, I was talking about the protein that is produced in this state which keeps everything repressed, that is known to be C1, and it's known that C1 can bind to different parts of the viral genome and prevent production of those genes and so on. And similarly, in the other state, it's known that C1 is low, CRO is high, and CRO prevents further production of C1. So it all seems to make sense, okay, at this level, at very hand waving level.
So C1 and CRO and what exactly?
They are proteins. So they are proteins, they are viral proteins produced by genes that are encoded on the viral genome. Okay, they are not bacterial proteins, they are the genes corresponding to these two proteins are encoded on the viral genome. So once the viral genome goes in, it uses the bacterial machinery to start expressing these genes. So then the proteins get produced, and there is a competition between these two, whichever one sort of wins the competition. So coming to the decision then you can imagine is something to do with, does C1 get produced first or does CRO get produced first? Whichever manages to get the upper hand and suppress the other, that's the way the decision will go. I'll come back to this a little bit more if I have time. Yeah.
Bacteria affect the virus's decision.
Yeah. So here's the thing. So this is a very simple sort of textbook picture, but of course life is more complicated. In the case of lambda phage, we actually now have the entire genome known. We have mapped out all the genes. We don't know all the functions of all the genes, but we know most of them, and there are many different genes. Some of them we know are produced early on in the lytic cycle, some of them are produced when you go lysogenic, some of them are useful, like these ones are useful for replication of the bacterial genome. These proteins are used to produce the final structure that sort of encapsulates the protein and makes new new viruses that can go out and infect new cells, and so on. And what is also known is a lot of the interactions. So if you look at what is known right now about the interactions between all these different proteins that are produced from the viral genome of lambda phage, there are still a few question marks, but there's a lot known. So this is the bit that I was talking about as the textbook picture. There is a repression of CRO by C1 and a repression of C1 by CRO, but as you can see, there's a lot of stuff going on. There's a lot of stuff within the viral genome, but there are also these red things, by the way, to answer your question, bacterial proteins that also affect the viral thing. So it's a back and forth. The virus is using the bacterial machinery, but the bacteria also affects which parts of the virus genome are expressed and so on. Yes.
How noisy are the expression levels of these other groups?
So they are quite noisy. It is a stochastic. So it has been measured in some cases. So C1 and CRO, which people focused on because they were the core, in the beginnings of the decision, they're quite noisy. And once the decision is made, the noise levels seem to be a little less. Okay. Again, maybe I'll come back to that question, but on the whole, you should totally think of this as a pretty stochastic and noisy system. This is not a deterministic system.
You expect the other proteins to be much more noisy than C1 and CRO?
Not necessarily. So a lot depends. Some are not. These two proteins are known to have a fair amount of fluctuations, but other proteins don't. Some of these other proteins are quite stable. These proteins that are involved in the lysogenic part are quite stable, and so on. So it's quite variable how much you actually see, and a lot depends in fact on the kind of, so one thing that affects that is the lifetime of these proteins. Some are very long lived and therefore they buffer against fluctuations in production because they are long lived, whereas others are very short lived and therefore they fluctuate a lot. So it varies a lot, but bottom line, do think of it as a stochastic system. Thanks, Razi. So, yeah. Sorry?
Is C1
Yeah. Under certain conditions. So it turns out that C1 is one of those proteins that can be both an activator and a repressor, but it also depends on what its concentration is. So at certain concentrations it acts like this, in certain concentrations. So you can imagine now the situation is a lot more complicated than that simple picture I gave you. C1 repress CRO, CRO repress C1, all very nice, up, down, two stable states, but it's clearly embedded in a much more complicated network. One of the things that, reasons why it's more complicated is because, of course, this decision is not just a flat decision which is just going to be lytic or lysogenic 50-50, throw a coin, or even a biased coin. A lot of the reason why there's a complicated network is because you are trying to pull in various kinds of information about the environment and bias the decision accordingly. So for instance, when DNA damage happens, this is quite well known, then just hand wavingly again, DNA damage is happening to the bacterial genome. This is not a situation in which the virus wants to go and sit dormantly inside a genome, because then you will also face the DNA damage and you will also get damaged just like the bacterium. The bacterium may die, you're sitting inside, you will also die. So it's known that in the case of DNA damage, when the bacterium senses DNA damage, the virus uses the bacterial sensory mechanism to detect DNA damage to jump out and make lots of copies as quickly as it can and get out. Okay. There are other things. Some of these detect things like temperature, poor nutrition. So again, you can imagine in various kinds of situations you might want to bias this decision. But this is such a complicated network, it's quite hard to understand actually how this decision, we don't fully understand. We know hand wavingly and in some kind of overall logic way, okay, when the bacterium is doing poorly, you should probably jump out. When the bacterium is growing nicely and growing well, has plenty of food, you should probably, you can, again, it depends. In some situations when the bacterium is growing exponentially, it would be a great time to jump out, because then you'll have many more bacteria to infect and you're never going to run out. But in other cases, it might be good to stay in. So it's a complicated decision. And I just wanted to leave you with that. And let's move to a sort of a different level to now ask, so this is sort of looking at it from a network perspective and looking at the subcellular thing. So can we zoom out and try and understand when and where a decision would be good to go this way or that way? And that's what we tried to do. So this is what I'll come to at this level. So one question of interest at this cellular level is, so how much is this? If you infect a colony or a liquid culture of say E. coli with lambda phage under certain conditions where say the bacterium is growing very well, what percentage would you expect to go lysogenic or lytic, dormant or virulent? It turns out that people have done a lot of these kinds of experiments with a wide variety of bacteria and viruses, and in these kind of conditions where the bacteria is growing pretty well, there's no shortage of food, it's in exponential phase, and you infect with a reasonable amount of viruses, you find that the lysogeny percentage, the dormancy percentage, is always between 5 and 15%, and this is across evolutionary very different viruses, very different bacteria, all temperate, all capable of doing this decision, but you find that it's 5 to 15%. Why is it not 25% or 50% or 60%? So different kinds of explanations have been put forward. One kind of explanation which a lot of people have made is that there are different kinds of conditions, the environmental conditions that a colony of bacteria and viruses will face. Some will be conditions where the bacterium is just in dire straits, it's dying, maybe DNA damage is happening, or there's some chemical which kills bacteria. In that case, you don't want to be inside, you want to be outside as a virus. So those would be conditions where you would want to jump out or be outside or produce lots of offspring and wait for times where you can find other bacteria. On the other hand, there are known to be other conditions where a bacterium is well insulated, not going to die. Any virus genome that's inside a bacterium is quite safe, but viruses that are outside will die. So both kinds of environmental conditions exist, and in a way this is like a bet hedging. You are going to sort of have some amount in this state and some amount in this state, so that whatever environmental condition happens, some of you will survive and can reproduce and rescue the whole population eventually. So in this case, the lysogeny percentage would be set by the frequency with which, the relative frequency with which you will get these two kinds of environmental catastrophes, where either bacteria would die or viruses would die. That would set this 5 to 15%. But we wanted to investigate another kind of alternative, which is to say, what if now you have multiple viruses attacking the same bacterial population, okay, and each one has its own percentage. Maybe there is one that goes 20% lysogeny, one goes 10%. If you have a competition between these two types of viruses for the same bacterial host population, which one would win? Can one talk about one of them winning? Which one would win? Would certain percentages do better than others? Okay, so I mean, imagine a situation where, for instance, you have lambda phage, which has a certain percentage of lysogeny, but you get a mutant which has a slightly different. So imagine these two are very, very similar phages, and the only thing different between them is the percentage at which how they do the decision and what percentage they go lytic or lysogenic. Okay, that's all, no other difference. So it's a bit of a hypothetical situation, but you can kind of imagine how it might arise from a mutant. So how do we investigate this? So in order to set up this competition, one simple way would be to set up a set of population dynamics equations, something like you may have heard of Lotka-Volterra type equations. So one uses Lotka-Volterra type of equations very commonly when one is talking about growing bacteria and viruses infecting them, because it is still a classic predator-prey kind of system, and Lotka-Volterra is one kind of framework people use. So for instance, if this is the uninfected bacteria, the rate of change of the population, there will be a term for the growth. This is a classic logistic term, but don't worry about the exact terms I'm using. Much of what I tell you will work even if we don't have a logistic term but just have exponential or have some other kind of growth. So people have tried different kinds of models for how to describe bacterial growth, how to describe viruses infecting bacteria. In some sense, what I'm going to tell you doesn't depend on very fine details of those. Anything that has at least a little bit of exponential growth, and then something like this where the rate of which you get infected is proportional to both the bacterial population and the phage population, which is understandable, just like a Lotka-Volterra predator-prey system. That kind of, those kind of terms will give you all the results that I'm talking about. So we can write down a set of equations for the set of bacteria that are not infected, the bacteria that are infected by the blue phage, the set of those bacteria that have gone into the dormant state. So L represents the lysogens, the lysogenic bacteria which have gone into those states. So I will be 1 and 2 over here. If you want to know the details, I can tell you more. But the bottom line is there are a bunch of parameters here. There are parameters like the growth rate of the bacteria. There's parameters like the infection rate or effective infection rate per phage of the bacteria. There are things like the carrying capacity. There are things like the rate at which the decision is made. So there is a certain amount of time it takes for an infected bacteria to decide to go this way or this way. When an infected bacteria bursts, then new phage are produced, which is set by this equation, and there is a burst size. This beta represents, so I'm just giving you a flavor of the kinds of parameters in these kind of equations. So these are parameters that you would choose numbers for depending on which particular pair of bacterium and phage you're going to use. If you're using E. coli and lambda, you'll use a particular value for beta, but if you use some other phage, maybe it's 10 times more or 10 times less. So these parameters can vary, but the overall structure is actually a pretty good structure. We know this kind of equations work very well with a single virus and a single bacterial species. People have done lots of growth experiments where they start bacteria growing and put in viruses, follow the growth curve, and you can reproduce all that and get pretty good parameter fits, which also can be independently verified, which give you all the growth curves very nicely. So this kind of Lotka-Volterra like equation generally describes a single phage and a single bacteria quite well. So we decided to try it out with two types of viruses, both infected. So here's typically what happens. Yeah, sorry. So I was kind of skipping through, but let me tell you in more detail. So B0 are bacteria that are not infected by anything. B1 and B2 are bacteria that are infected by either phage type 1 or phage type 2 but have not yet made the decision. L1 and L2 are bacteria that have gone into lysogenic state. They are either lysogens of type 1 or type 2. They have been infected by phage of type 1 or type 2 and that phage has gone dormant. So they can't be infected further, as we discussed earlier. P1 and P2 are the number of phage that are outside bacteria, the free phage. Those are the ones that actually infect uninfected bacteria and infect them. Okay, so the rates of infection depend on the phage population that's outside, which are the P, and on the uninfected bacteria and things like that. Okay, is that clear?
So what is F and
So these are again parameters. Okay, so let me define F. So I told you about this burst size. The F is, so think about this. There is a certain number of bacteria of type 1 infected by phage type 1. There is a term here which represents the decision. So there's a certain rate at which bacteria of type 1 effectively either become lysogens of type 1 or burst. So this delta B1 will split into two terms. One will be a certain fraction, F1 will go into the lysogenic state. So that is the percentage that go lysogenic upon infection. That is what I was talking about, this 5 to 15%. The rest of them, 1 minus F1, go into this burst state and produce a burst of beta new viruses. Okay, so in fact this is a pretty crucial thing. This 20% lysogeny or 10% lysogeny will go into the parameters F1 and F2. Okay.
So PI are like free viruses.
Yeah, these are free viruses. These are free viruses sitting outside. So PI, you can think of these you can think of as B0, and these are B1 and B2 which have not yet, and I'm not representing the lysogens here. You can think of these as lysogens if you want, and somewhere in between there is this temporary state of B1 and B2 which will either go and produce more of these or go into the lysogens. Yeah.
All the parameters are comparable for both the viruses.
So this depends on what you're exactly trying to model. In the scenario we wanted to understand, we were trying to think, what if everything is the same and the only difference is this decision, because we want to understand what makes the decision 10% or 20% or this number between 5 and 15%. Is there a reason why in a competitive scenario it would be optimal to do something between 5 and 15% and very non-optimal to do something like 80%? That's the kind of question we want to get at. So indeed in the simulations that I'll show, I will keep all the other parameters the same for both but change them. But of course in any other kind of realistic ecosystem you'll have all sorts of viruses, and so imagine this is more suitable for a scenario where you think of one virus that is sort of adapted to a particular population and then a new mutant comes up which has everything else the same but has just changed the way it does the decision a little. In that scenario this might be a relevant thing. Yes?
So P1, like the free viruses can't grow by themselves, right?
No, free viruses cannot grow by themselves. They can only grow by having one of the B1 or B2 decide to burst. So this term comes only from bursts, and this comes from infection. So when you infect, your DNA goes in, and if it doesn't come out, then it's gone. So there is a negative term that sort of is the adsorption of viruses into bacteria, and if they go into the lysogens, then they don't come back and promote the P1, but if they burst, they come back. Okay. So yeah, I was not sure, thank you for asking these questions, because I was not sure just how much detail of these equations to get into, but for those of you who don't want to get into too much detail of the equation, the bottom line is it's a predator-prey system, and it's just one way of modeling a predator-prey system, and I'm sure you've seen other ways, but pretty much any predator-prey system will give you the kinds of results that I'm going to tell you about. Okay, we have tried other things. So let me give you a scenario, an example of what happens in this dynamical system to the populations over time. If we start with a very small number of bacteria, we're starting with bacteria in its growing exponential phase. There's plenty of food for the bacteria to grow, and you infect it by an even smaller number of viruses. A tiny number of viruses are around, and they'll slowly start infecting and then doing this process of infecting and bursting and so. So this is an example exactly for this situation where one of the viruses has 20% lysogeny, one has 10%. So initially the bacteria grow almost as if the viruses don't exist, because the viral numbers are very low, hardly any bacteria are being infected. The viral numbers are really, so the dashed lines are the viral numbers, the solid lines are the bacteria, and just think about the blue curve, which is the total bacteria for now. So initially there's exponential growth as you expect, then at some point the viral numbers start rising very sharply, okay, and at that point you can see a dip in the total bacterial numbers. And this is because bacteria are now dying. There are lots of viruses around, they're infecting a lot of bacteria, they're killing them, lots of them are bursting, some of them are going dormant also. But a lot of basically what happens is very quickly all the healthy bacteria, that is the ones that are uninfected, they crash. They go from being whatever number they were to really crashing very quickly. And this is because the virus numbers go up very fast. You can kind of imagine why this is happening. As I told you, the way the virus works is, each virus infects a bacterium. It's like somebody was asking, right, can one virus wipe out a whole colony? Yes, it can, because the viral numbers can really shoot up very quickly, because that is itself an exponential process. There's each virus infects, produces say 100, which go infect 100, which produces 100 more. It's exponential, right? But the bacteria themselves are growing exponentially. So if you look at the equations and try to understand how this curve looks for the virus, it's like a super exponential. It's an exponential on an exponential, which is why it's very flat for a long time and then suddenly shoots up, which is why the bacteria grow exponentially, pretty close to exponentially for a long time and then suddenly crash. So it's really like two phases. There's bacteria almost unaffected until suddenly the virus population shoots up and the bacteria population goes down. But at this point, because there are certain percentage going lysogens, it's not that even if the uninfected bacteria crash, it's not that all the bacteria are dead. There are all these lysogens still around, and those lysogens, as we discussed, are immune to further infection. So now they start growing exponentially. There's really nothing to stop them. There's still plenty of food. So then a new exponential growth kicks up, which is shown here. These are the lysogens, and the lysogens start growing and the bacteria eventually come back and reach carrying capacity, okay. So in all this dynamics for these equations, if you start from a small number of bacteria and a small number of viruses, you eventually reach a state where bacteria have reached carrying capacity, all the viruses have kind of been absorbed, all the free viruses, and all you're left with is lysogens, and this entire carrying capacity is made up of certain fraction of this and a certain fraction of that. And now you can kind of declare a winner. You can say, in this state, that's it, that's the end of the game. The bacteria have reached the maximum they can, and they are split into two types, this one or this one. So whichever is higher is the winner. You can even try and quantify it. You can make it a percentage between -1 and +1 to quantify by how much it's won, okay. So in this case 10% beat 20%. So this is now where we can bring in some game theory. So what we're doing is we're kind of taking this whole dynamical system and thinking of it as a game. The two players, it's a two-player game. The two players are the two types of viruses. We need to define a strategy space. Here the only thing we're allowing to vary is the lysogeny propensities. So the strategy space is the space of possible values that you can take. Your strategy is, as a player, I'm going to do 10% lysogeny, or another player will say I'll do 15%. Let's say we stick to these possibilities, a fixed percentage. If you make the strategy space such that it's basically a fixed percentage of lysogeny, you can imagine building up a payoff matrix. I guess now, I hope you've heard through this workshop enough of game theory to recognize some of the terminology I'm using. So imagine these are the strategies of the second type of phage, and this is the strategy of the first type of phage, right? So the strategy, technically it's a continuum, you could use all, but in practice what we'll do is we'll split it up into lots of tiny 0.01, 0.02, 0.03, and then play against 0.01, 0.02, 0.03. But you can imagine making a large, maybe even continuous payoff matrix where the values of the matrix tell you did phase 1 win and by how much. Okay, so the values of the matrix, the payoff is the payoff for phage type 1. So phage type 1 strategy, phage type 2 strategy, and what is the payoff for phage 1. These are imaginary numbers I've put in, but basically something like 0 would mean a draw. In the dynamical system, if both viruses ended up at 50-50, then that should give you a payoff of 0. If phage 1 won and completely wiped out the other, that should give you a payoff of +1. And if phage 1 completely got wiped out and phage 2 was up, that would be -1. So you can fairly easily from this dynamical system, from the end state, from the final stable state, convert it into a payoff matrix. And you can run each strategy pair against each other and build up this entire payoff matrix. Yeah.
But in that case, I will anyway have to do all the dynamical simulations
Yes.
beforehand.
Yes,
So
yes.
then I already know.
Yes. So here what we're trying to do is, given this dynamical system, if we do all these dynamical simulations, can we figure out is there an optimal, and if so, what is the optimal?
But by doing all of this,
By doing, by doing. I mean, so later on I'll show you can do a little bit of analytical work to get to this, but to begin with I just wanted to show you brute force. We are just going to run all these simulations, right? And he's right. I mean, in the end, if you wanted to know, you would just run the simulation and it would tell you. If you wanted to know, will 10% do better than that or whatever. But yeah.
the payoff, what exactly is the payoff? Like usually it is fitness.
Yeah, so here it is a kind of fitness in the sense that in the final population, which one has the larger population? Which lysogen, because we only have lysogens at the end, which lysogen? So you can call it a certain kind of fitness.
In this case, will you call the lysogen the payoff of the
Yeah.
10% like towards 0.5 further and that?
Yeah, a little more than 0.5. I mean, if it were 50-50, I mean, the payoff would be 0. So here the payoff for this, if it was phase 1, would be, I don't know exactly what value it'll get, but it'll be plus something. And if this curve was below that curve, it would be a minus. So indeed, I mean, the payoff is just a normalized. It's a zero-sum game, absolutely. So indeed, so here's actually a picture of what we get by running all these simulations. So indeed, it is a 2-player, zero-sum, symmetric game. It's completely symmetric also. So this is the kind of payoff you get if you're plotting the payoff for phage type 1 as a function of the strategy space of player 1 and the strategy space of player 2. And as you expect, I mean, it need not be like this, but in this case, everything is based on a fairly smooth dynamical system. You get a nice saddle point. And this saddle point is exactly what one would identify as one kind of best strategy. You've heard of many ways of, I think, defining the optimal strategy in a game, right? You've probably heard of ESSs, as Sagar was talking about. You've heard of probably Nash equilibria. You've probably heard of minimax strategies. So in fact, this is the kind of thing you would identify if you did a minimax algorithm on this. What does a minimax do? You keep the strategy of phage type 2 fixed, let's say you keep it at 0.5, and you check all the other strategies and you find which strategy would be best for one. The one that would be best is the one that has the highest number. So for this strategy of phage 2, this would be the strategy for phage 1 that would be best. And you do this for every single phage 2 strategy. And you find all these best strategies. And then you ask, okay, of all of these, which one would phage 1 pick? Which strategy should phage 1 pick? And it would pick the one that would give you the mini max, right? It would be the minimum of the maxima. These are the maxima you can get, and this would be the minimum. Yeah, sorry.
I'm a bit confused. You said that these different strategies are the different fractions
Yes.
of lysogen.
Yeah.
But you also mentioned that the lysogeny fraction lies between 5 and 15%.
That's the observed.
Yeah.
The observed in experiments lies between 5 and 15%.
But here
Here we allow
extending beyond.
Here we allow 0 to 1. I mean 0 to 100%. So what we want to do from this is, in a competitive scenario like this, would we get 5 to 15% coming out naturally? Okay, so this, by the way, is simulations run with a particular set of values for gamma, k, eta, and things like that. The only thing that changes, everything else is fixed. Like beta, I remember was something like 100. I've forgotten the values we used over there. But the only thing that changes is f1 and f2. So these are f1, f2, and here again these are f1 and f2. And for each value of f1 and f2, you run this dynamical system, you see who wins in the end, you build basically this payoff matrix, and you find there is a single minimax strategy. You can see what the minimax strategy is here, why it looks like a saddle point, right? Basically, phase 1 is trying to maximize its payoff, this is the payoff for phase 1, and phase 2 is trying to minimize it. So what is the strategy they will settle to? The strategy at this saddle point, there is no incentive for individually either player to change their strategy, because if you're sitting there and player 2 changes their strategy, then player 1 can easily find another strategy that's a little bit better than the saddle point. Similarly, if player 1 changes a strategy, player 2 can always find something that goes down a little bit because you're on this saddle, right? So this is really in this kind of 2-player zero-sum symmetric kind of game, the saddle point is naturally the kind of place, if it's accessible. That was of course another thing that Sagar was talking about. If that is accessible and evolutionarily you could reach it, there would be no reason for individually any player to move away from that. Okay, so in this case, because it's a pretty simple sort of game, the Nash equilibria, the ESS, the minimax, all coincide. They're all the same. They're all there. Okay, now it turns out as you can see, this saddle point is around 10%. Okay, it so happens to be close to 10%, which is within that experimental range we found. The numbers we took and used for the parameters were sort of numbers that are known for lambda phage. So let's get back to that in a second, but let me just give you a sense of why there is an optimal in the first place. It's not so hard to understand why there's an optimal. As you can see, if we look back at what is the trajectory of these typical dynamical systems, as I said, the trajectory is first the bacteria grow pretty much exponentially nicely without much, and then they crash very quickly. So the game really ends already here. Why? Because at this point there are 0 bacteria that have not been infected. There are a small number of lysogens of each type, and there is absolutely no healthy bacteria left anymore for anything to infect. So all the viruses that are hanging around outside, they don't have anything to infect. They are just out of the game. What happens about who wins here is actually decided by how many lysogens are here at the point where the uninfected bacteria go to 0. At that point the game is over. So what you really need to do is to have more lysogens at this point. In order to have more lysogens at this point, it would be nice if you went lysogenic in every infection that you did in this phase, right? But the problem is, if you did lots of lysogenies right from the beginning, you wouldn't have a very high population out here in the first place. So there's a pull in 2 different directions. You want in this crash part to have lots of things go lysogenic. But if you make yourself a virus that always goes lysogenic a lot, then you won't even build up much viruses in this phase to even infect at that point. So it would be perfect if you could go lytic all the way and then switch to lysogenic. But in this game we are forcing them to pick a certain percentage. So they are pulled a little this way, pulled a little that way. The optimal ends up being in this case 10%. That's not surprising in a way that there is an optimal because of this pulls and pressure. What is a bit surprising is that now if we rerun these entire game with completely new parameters, not parameters that are set for lambda phage. So what I'm showing here is the final the optimal lysogeny percentage that comes out of this game theory sort of thing, this payoff matrix and all that, but for different parameter values. So we changed the burst size, the decision rate, the number of initial bacteria, the growth rate of bacteria, the number of initial viruses, the rate of infection. All of these we changed by many orders of magnitude, and it turns out that you sort of are always in this range, very few. Only in this case where you have a case where the number of initial viruses are very large, that case sometimes you get an optimal that goes up to about 30%, but actually you're stuck between 5 and 15%, which is exactly what we see. That's a little harder to explain. It's not obvious. We can do a little bit of analytical stuff to try and show, basically if you make the assumption that this kind of crash is instantaneous, you can take those equations and do all the game theory without running the simulations and get an expression for the optimal. And we find the reason why the optimal does not depend too much on parameters is because the parameters come into the optimal, the expression for the optimal percentage in a log, inside a logarithm. So large changes of parameter values make only small changes to this logarithm and don't change the overall value too much. That's a mathematical explanation. Physically it's a little harder to explain, but it's something to do with the fact that the parameters that really matter are parameters that affect the phage efficiency, how good the phage is at infecting. And it's sort of,
Is that something that you get from the figure, or you do it from some other
from the equations. From the equations with one assumption that this crash is practically instantaneous. So if you make that assumption that the crash is instantaneous, you can analytically do the whole game theory, you can find the Nash equilibrium, and it turns out to be this. So it's something to do with the fact that phage efficiency also pulls in both ways. If your phage is very efficient, you will start rising very quickly, but the phage, the bacteria will also not be able to climb very quickly. And it turns out that no matter how much you push and pull the parameters, you end up sort of seeing a curve that's very similar. This dynamics does not change too much in terms of the time scales. Usually it takes about 1 or 2 bacterial generations for the bacteria to grow, and then a crash that happens very quickly. So somehow the fact that whatever kind of virus you use, less efficient or more efficient, the dynamics is not perturbed too much is part of the reason why you end up having a very small effect on the final optimal percentage. Okay.
How you telling that from this figure that it will be beta and
Yeah.
P0, which would be the deciders? Like, how would I read this figure?
No, so in some sense, even beta does not really affect too much. It's still within the very large, the very tail end here of P1 is what seems to affect, and that is partly because at that tail end this number actually becomes this becomes a little larger, and this log just drops out. I mean this effectively becomes close to 0. It starts to go close enough to 0 that it works out. You could actually see the same effect with eta also. So actually any of these could show the same effect at P0, except what we're doing is we are looking at sort of the lambda phage numbers, which are these dark lines, and looking at ranges around those. If you extended the range of eta even more, then you could get a situation where you would push this to 30. But so this is the lambda phage. The darker region is sort of what is known for other phage and is what one would call a biologically reasonable range, and the rest of the range we went a little bit further than the biologically range. So the point is that for P1 you could imagine this range and pushing it to 30, but for the adsorption rate we're so far from the biological rate that it didn't seem sensible to even think of that. But if you do push it that far from the biological range, you could get this to about 30. But beyond that is very hard. Beyond that, and that you can tell from the log, that you wouldn't get it beyond that.
Beta is not inside a logarithm.
Beta is not, but it's in a 1 over beta form. And beta is usually, to get beta to matter, it would have to be small. And usually beta is in the range of if you bring beta to about 2 or something, then you would start to matter, 2, 3. But usually beta is at least a factor of 10. Once you're a factor of 10 or more, usually it's actually a factor of 100. Then it's out. This is just too small compared to 1. It doesn't matter again very much. So we looked at 2 to here, and in 2 you can see it starts to push. When you're down to 2, 3, 4, you're starting to push this a little bit. But as soon as you're 20, 100, 10 to the 3, it's all in a very narrow range. So basically here it doesn't matter because you really need to be 2, 3, 4. You imagine that's a phage which only produces 2 more phage when it infects. It's losing 1 when it infects. It needs to produce at least 2 to even break even. So below 2 anyway the virus is dead. So 2, 3, 4 is not very likely. We've not found viruses that go down to such low numbers. Okay, I think I have maybe about 5 minutes left. So let me go from this. Yes. Yeah.
So when you were saying it's related to weight hedging,
Yeah.
you meant diversified weight hedging? Or?
That was different. In a sense, that's a different explanation people give for the same phenomena. Why is it 5 to 15%? So some people have said, no, it's because the environments that these viruses face are so variable that sometimes there's an environment catastrophe happens to all the bacteria. Sometimes some chemical comes in and all the free phage will die, but the bacteria will survive. So to survive these catastrophes, you need to be some percentage in this, some percentage. Yeah.
Is it diversified weight hedging or the adaptive point of view?
Maybe more like the diversified, but you know, I mean, again hard to say. There's some conditions in which I can imagine virus bacteria games which are more like the adaptive pointers. But you know, I mean, nobody knows how these catastrophes, how often they happen. So that explanation is a little bit just pushing the explanation. It doesn't give you a real explanation for why this number. It just says this number is because the environmental relative ratio is this number. But nobody knows why that should be 5 to 15% either, right? So we are proposing an alternative which is less to do with the environments and more to do with competition. Yeah.
So when you're weighing these parameters and mapping them onto the lysogeny fraction,
Yeah.
so you're weighing one at a time.
Yes. This is varying one at a time, but we have also done very multiple and many, and you know, within this ranges that I've sort of shown you, and basically
At a time, the others are fixed at
the others are fixed at the lambda phase, the dark values.
the dark line,
The dark.
solid line.
Yes. Yeah. So in this figure, yes, but we've also done a little bit more of a. This is easier to see. That is hard to visualize. So we've also done where you vary all of them randomly within the ranges, pick totally randomly within this hypercube, and there again, you know, you find basically the same kind of thing. Okay. So there's only one more point I want to make, little bit of a teaser. I'll go through this a little quickly. Right at the beginning, I told you that, you know, of course, this whole picture is more complicated. There are all these bacterial proteins that are also involved in the decision, and it's all because maybe there is a way of getting information from the environment, right? And different kinds of information comes in and affects the decision. So let me give you one example of that, and I'll stop. One very interesting case is where there are multiple infections. So you can sometimes find that a bacterium will get infected by 2 individuals of the same type of virus. Okay, sometimes it'll get infected by 1, but sometimes 2. So somebody actually did this experiment back in the 1970s to see whether this has an effect on lysis lysogeny, and it does. So each of these dots is a different experiment, an experiment in which a certain ratio of phage and bacteria were taken, mixed together in conditions where they would not infect, but they would just attach onto the bacteria. Okay, so let's say at 1:1, average phage input 1 means 1:1, 10 bacteria, 10 phage were mixed together. They would all randomly attach and absorb, and then he would change the condition and they would all infect simultaneously. So what happens in that kind of case? The certain percentage, then he would measure how many went lysogeny, what percentage, and he did this with different ratios. So he's starting with ratios where there are many bacteria and very few phage, and a small number goes lysogenic. But if you go to a very large number of phage to bacteria, 10:1, then almost all of them go lysogenic. So clearly the lysogeny percentage is now not 10 to 15. The 10 to 15 number comes here in this close to this 1:1 range. So we were talking about 10%, 15%, that's this. But if you now vary the ratio, it varies all over the place. So why does this happen? So when you're in the way this experiment was done, as I said, they first infect and they sit. So some bacteria will escape. By pure chance, no virus will attach to that bacteria. That's called a multiplicity of infection 0. Some will get infected by 1, again pure chance. That's called multiplicity of infection 1. And some will be multiplicity of infection 2, 3, 4. Depending on the ratio you start with, you'll have different probabilities for these, right? and you can kind of guess that the way this will be distributed is a Poisson distribution. In fact, this guy was pretty clever. He did a whole series of clever experiments to prove that it is a Poisson distribution, but I won't go into that. But you can believe that it's a Poisson distribution. So you take a certain number of viruses, a certain number of bacteria in a certain ratio, mix them together and randomly distribute, and you'll get a Poisson distribution of how many viruses have how many bacteria have 0 viruses, how many have 1, how many have 2, and it's a nice Poisson distribution. Knowing this Poisson distribution, you can extract what he has measured is this final number, the percentage of lysogeny given a ratio of phage to bacteria. What we know is this is Poisson. What we are out to get is this. What is the probability that this will go lysogenic? What is the probability that this will go lysogenic? And it turns out to be not the same. So if you look at the data and you fit it with different probabilities, I'm not going to show you a very complicated fitting and all that, but a very simple rule comes out. A very simple rule explains the data pretty well. And the simple rule is so this is by the way for E. coli and lambda phage. If E. coli bacterium gets infected by a single lambda phage, then it almost always goes lytic. It almost always makes lots of copies, kills the cell. If it is infected by 2 or more simultaneously, it almost always goes lysogenic. Okay? So this is a way, and the way people have sort of talked about this again hand wavingly is, this is a way for the virus inside a bacterium to make some estimate of what is the conditions outside, what is the ratio of viruses to bacteria outside. If the ratio of viruses to bacteria outside is large, the chance of having 2 or more simultaneous infections is large. If the ratio is very small, you will mostly have a single infection. So it is trying to guess the conditions outside and adjusting the decision accordingly. But forget that hand waving explanation. We can take our same equations here, and instead of having an equation with a fixed Fi, we can modify these equations to also allow the dynamical system to have multiple infections. We after all gave a parameter delta where there was a certain time before before the decision happens. So if another infection happens in that time, we can allow it to be present. The equations become more complicated because you have to keep track of how many are infected by 1, how many by 2, how many by 3, and you have to give new parameters. So now the strategy space is also much bigger. It's no longer what is the percentage for this phage and this phage. It's this phage with multiplicity 1, this phage with multiplicity 2, and you can allow all sorts of strategies, right? So of course this is a huge strategy space. It's kind of hard to explore this completely either analytically or even numerically. So one of the kinds of things, and you would have heard about this in various talks, is you can do a kind of an evolutionary game. You can actually run a little evolutionary simulation and say let a population with different start off with different strategies, and whichever strategy is doing a bit better reproduces, and in the next round of the game, let it expand. So effectively you run something like Sagar was talking about, these replicator equations. Effectively it becomes like a replicator equations on this strategy space. And what we find, interestingly enough, is the strategy that really comes out is exactly this one, that if you go lytic, if you have a single infection, you should really go lytic. If you have double or more infections, you should really go lysogenic. This strategy seems to beat out all other strategies, including those fixed percentage 10, 15% ones. And again, you can kind of go back to this and understand why. I said that the best thing to do would be really, if you could know when the crash happens, the best thing to do would be to go lytic all the way here, and then as soon as the crash begins, you should go lysogenic. Because this way you maximize how many you build up to be around in the environment, so that when the crash happens, you take a large fraction to yourself. Now, it's impossible to know exactly when this crash will happen, but this strategy of looking at the single or double infections turns out to be very close, a very good way of predicting this point. And it's because the number of phage are very small here, so the number of infections, whenever you have an infection, it's almost always a single infection. The chance of double infections is very small here. And when you're here, the number of phage is shot up and the number of bacteria are starting to slide. So very, very often you get multiple infections. So there is a little bit of error you'll make very close to the crash, little bit before and little bit after. But compared to most other strategies, unless you had a strategy that could really detect a peak and go down, which we don't know of any such strategy that can do that, this comes very, very close to detecting this transition very well, and therefore it beats everything out. So, I mean, let me just summarize. What we have tried to do is use a combination of dynamical systems and game theory to show that firstly, there is reason to believe in a competitive scenario, there is an optimal percentage. There is pulls and pressures in two ways, because in a competition you want to produce enough offspring to basically take over the lysogens, but you need to go lysogenic also. So you're being pulled towards the lysogenic to have enough lysogens eventually at the end, but you're being pulled towards the lytic to have enough in the middle when the crash happens. So these two pulls and pressures basically give you an optimal. But what's interesting is that if you really see the dynamics and look for really what should be the optimal, a very good way of approximating this real true optimal strategy is to look at multiple infections and to base your decision on whether you have a single or a double infection. And we find that a lot of phage, lambda phage we now know does this, we have good proof, but there are lots of other, now people are doing the similar experiments with other kinds of temperate phage, and there is more and more temperate phage we know are doing this lysis-lysogeny decision based on this number of infections. Okay. There's a whole class of questions about how you would set up a network to do this, which I don't have time to go into, but it's also something we've studied. But you can now think of lots of interesting questions. How would a virus, set up a gene network which can be bistable, but can also change the final outcome, which stable point gets picked depending on whether it starts with a single genome or two genomes. And it's interesting to ask that kind of question also. So let me leave you with that. And if you have any more questions, thanks very much for your attention. Yeah.
I didn't follow on the first set of equations which you showed. Was it over multiple generations or single generation?
No, the first set of equations just is basically gives this. Yeah.
Trying to understand the population change and all that,
Yeah.
VIP and all.
Right.
Where there were multiple generations of viruses or single generation.
This is so these equations, the simplest ones, these is just, you just start it with some initial conditions and run it
You're done. Okay.
until it reaches steady state.
My question basically is the kind of game you show.
Yeah.
Of course, in that game, Minimax, Nash, and ESS all coincide. Why do you have a problem call Minimax and not ESS or not Nash? Is there any reason? Because if it's multiple generation, I would go with ESS.
No, yeah, I totally agree. In this case, they were all the same. It was more for an explanatory sort of thing. It's just to explain how would you find this.
True. But if you call Minimax,
Yeah.
suddenly I start thinking vectors are rational and all that.
Well, yes. So they're not rational, but on evolutionary timescales, you could kind of argue that yes, that they are trying to find whichever strategy beats, and so actually, ideally, what you would do is in this kind of simple game, we kind of know that if we did run an evolutionary thing over multiple generations, because it's such a smooth surface and it's so simple, you would only end up there. So it's all accessible and all. In a more complicated scenario, I would agree you would really need to figure out, can you access that state or not, which is the state that you will actually evolve to if you did a multi-generational thing. So the real bacteriophage thing is of course a multi-generational, many mutants, new mutants have arisen and you take over or not take over and so on. This is a very simple scenario to kind of, it's only more like a proof of principle, partly to say there is an alternative to the bet hedging explanation. Since the bet hedging explanation was the only explanation that seemed to be around for these decisions, we wanted to sort of present another scenario which says you can also think of a competitive scenario. Now it would be actually really interesting to set up more realistic competitive scenarios to see if the same kind of thing happens. Yes.
Is there an upper limit on the number of bacteriophages that can infect bacteria and go lysogenic?
Not yes. In terms of the kind of experiments that have been done, see, there is an upper limit in the sense that physically there's an upper limit because the way these viruses work, they attach to receptors, protein receptors that are on the surface, of which there are a finite number. They're quite large, but they are still a finite number. So, you know, anyway, once all those receptors have been attached to by viruses, you can't attach anymore. That number is probably in the hundreds though, and I don't think anybody has actually done experiments with that level, because as you saw in these experiments, already by 10:1 you're already hitting 100% lysogeny. So nobody really went much beyond 10:1. You might find some conditions where people are growing bacteria and all, where 10:1 happens naturally or thing, but so experimentally nobody has gone beyond 10:1. Physically beyond 100:1 there won't be anything, but we have no reason to believe it before that it comes down at all. There's no real reason to believe that. There is some evidence, a little bit of evidence, that as you go further here in this region, the decision slows down, that by having so many viruses sort of inside and, you know, trying to replicate and so on, the decision, the use of the machinery of the cell and so on, just the whole process slows down a little bit. Not by much, by a factor of 2 maybe. So, you know, there are a few things that happen when you really start stuffing, but I mean anyway the virus is handles a situation where anyway it produces usually about, you know, few hundred particles. So I would imagine up to a few hundred there would be no issue, and beyond that you'll never get, because you'll never have receptors that many. Does that answer your question? Oh, sorry, yeah,
for the lytic state, you need the genetic element of the virus to be integrated, right?
for the lysogeny, the dormant state, yes.
For the dormant state.
Yeah.
So is that a bigger constraint? How many particles it can?
So indeed, once one is integrated, the others cannot. So what happens is basically they get diluted out as the bacteria grows and divides. So it's not that they go away, but, you know, the bacterial population keeps growing and they don't replicate. So in the end you only have, you know, in the population you'll only have one. So if you're following the bacterial population, over time you'll see that all the lysogens have just one integrated and nothing else floating around. So in a sense it doesn't seem too much of a constraint. Whichever one, these are also situations in which we are talking about a single type of virus, multiple of them infecting one. So whichever genome integrates doesn't matter. Somebody asked about competition, there are known cases where multiple types of phage, if they infect simultaneously, there is a little internal competition about who can integrate first, and whichever one can integrate first kind of wins. Different kind of game which I have not really looked at. Yeah, well, let me finish.
Bacteriophages are DNA viruses?
What I'm talking about are DNA.
So all of these only holds for DNA viruses?
This only holds for DNA viruses. I mean, some aspects might hold for other kinds of viruses, but people have not studied it in such detail enough for me to confidently say which aspects will hold for this. Yeah, sorry, one question back, then I'll come back.
Cell cycle duration be affected by the bacteria? The cell cycle duration.
So typically what happens is, once the infection happens, the cell does not divide. Here. The cell, you typically rarely see the cell divide, I mean unless it goes into the lysogen. Once it goes into the lysogenic state, then it's dividing normally. But in this decision period, there's usually not much growth, and sometimes you see a little growth. You do see the cells elongate a little bit. So you see some growth but no division. The time scale on which this decision happens is in fact usually a little bit faster than the cell division time. So, you know, in some sense it's not, I mean, in some sense, I think the best way to say it is that cell growth and division is stopped as soon as the virus starts using the machinery. Unless it decides to go here, then it's restored. So there's not a big change. Once you start dividing again, there's no change. It's back to normal. Here, basically, there is no division. So it's in infinity. Sorry.
So, what exactly is the mechanism of double infection? Like, is it like one has infected and is in the lysogen?
No. So the double infection, what I meant is just two genomes get in simultaneously or very close by. I mean, now people have done experiments where a second genome, see there are multiple, again I said there are multiple receptors. So one virus attaches to one receptor, one attaches to another, and stochastically this infects, and very soon after, if this also infects, you have a bacterium which has two viral genomes inside it, both of which are producing viral proteins. So that's it. A double infection means that.
Already infected viruses will be
No. So what happens is, if you're already infected and well into this state, then if a second comes in, nothing happens. It stays in this state and nothing happens. So we're talking about double infections at a very early stage and very close to each other. And that only really happens when there's quite a lot of viruses around, so that the probability of that event is high. Yeah.
So you said that only one virus can integrate into the bacteria. So how does a multiple lysozyme?
So that happens because different types of viruses have different sites in which they integrate. So lambda-phage integrates at a particular place. It has something that will recognize a particular site, cut it open and put itself. Another virus that will also come into E. coli will have a different site where it will cut and go. So the multiple lysogenies happen because they're all in different sites. And typically for viruses that we know of, like lambda, there's typically only one site. Sometimes there are a couple of sites where they can integrate, but like for lambda-phage, there is a single site in the E. coli genome where it will integrate. It will not integrate anywhere else.
If there's double infection, both the like the genome of both the phages will be at the same.
No, only one can go in. The other will just float around. And there is this protein C1 which is also floating around, which is keeping both genes repressed. The one that's integrated as well as the one that's floating. Now you imagine the bacterium doubles and divides, then there is one bacterium which has one floating still and one which has none because it's divided. So as the bacterial population grows, more and more have only the one integrated and nothing else. And there's one bacterium floating around somewhere which has one extra one floating around. So in terms of the whole population, very soon 99.99% of the population just have a single integrated one. Any other leftover ones are just diluted away. Yeah.
So is that a lysogeny happens when the same kind of bacteria, bacteriophages infects the attaches to the bacteriophage, or
so the double infection part I was talking about, that is for the same two genomes of the same bacteria. Those experiments are all for the same bacteria. Yeah. Same bacteria, same virus.
Same kind of virus.
Same kind of, exactly the same virus. Not even same kind. But people have done experiments with closely related viruses. So if the viruses are close enough related, then simultaneous infections are just like if they were identical. But if they are a little bit further apart, they're not evolutionary as close, then different games happen. Then competition can happen inside between those two. All sorts of games are possible.
Can we do this scenario something like when there is some lysis happens? So the nearby bacteria will get more diffusion, more.
No, absolutely. Good question. And this kind of brings us to this scale of question where, what are the patterns at a level of a population. So indeed, and now actually my colleague Shashi Tuttupalli has experiments where he's in fact imaging viruses bursting out of lambda-phage, bursting out of E. coli. And you can absolutely see that when a burst happens, there is of course a certain spread. So very close by you get double infections, little bit further away you get single infections and so on. And the range to which this burst happens, so it's not quite as simple as that, because the burst actually turns out to be not just a passive burst, it's actually an explosive burst where the phage come out of the poles of the bacteria. The bacteria is actually more rod shaped. So they come out of the poles and they explosively are shot out. So it's not always just close by. It's asymmetric, it's not, it's anisotropic. It's not like in all directions you have the same probabilities. So there are more complications now at a realistic level, which we don't understand the implications of that.
Yeah. The falling phase.
Yeah.
There will be like lot of frequency of this burst.
Yes.
So there could be like high chances of getting double infections and going to polygenesis.
Absolutely. So it seems, and this my colleague does see, that when you have a lot of bursts happening near each other, the bacteria around which get infected tend to go lysogenic. Now what are the consequences of that to how the virus is spread through a colony, and what happens when the colony is also growing and all that is still up in the air. We have, he has nice pictures and beautiful spatiotemporal patterns, but we don't understand them really fully.
With that, solve this because it's coffee outside.
Thank you so much. Thank you for all the questions.